In this video, we will discuss CRISPR's role in genetic engineering. CRISPR, clustered regularly interspaced short palindromic repeats, technology has rapidly emerged as one of the most impactful tools in genetic engineering. Originally discovered as a natural defense mechanism in bacteria to fight off viruses, CRISPR has been adapted for use in various scientific fields as a powerful gene editing tool. It allows for highly precise, flexible, and relatively efficient DNA editing in living organisms, enabling scientists to alter genes in previously unimaginable ways. CRISPR is paving the way for new advances and possibilities in genetics from medicine to agriculture and conservation. 1. CRISPR in medicine, treating genetic diseases and beyond. One of the most promising areas for CRISPR's application is in the treatment of genetic diseases. Many inherited conditions, such as cystic fibrosis, sickle cell anemia, Huntington's disease, and muscular dystrophy, result from mutations in a single gene. CRISPR offers the potential to directly correct these mutations by cutting out the faulty gene sequence and replacing it with the correct version. Gene therapy for genetic disorders. CRISPR could enable gene therapy for genetic diseases by targeting and fixing specific mutations. Clinical trials have already shown positive results in treating blood disorders like sickle cell anemia and beta thalassemia, where CRISPR edits blood cells to overcome the genetic defects that cause these conditions. This approach could extend to other single-gene disorders, offering long-term relief or potentially even cures for patients. Cancer treatment. CRISPR is also being used in cancer research to enhance immunotherapy. By modifying a patient's immune cells, such as T cells, CRISPR can help these cells better recognize and attack cancer cells. Scientists are exploring ways to reprogram immune cells with CRISPR so they are more effective in combating various types of cancer, offering new hope in the fight against this complex disease. Infectious diseases. In addition to genetic disorders and cancer, CRISPR could potentially play a role in fighting infectious diseases. Researchers are exploring the use of CRISPR to target and disable viruses, like HIV, by editing out viral DNA integrated into human cells. This approach may help develop therapies that go beyond traditional antiviral drugs by directly eliminating the virus at the genetic level. 2. Agricultural advances with CRISPR. CRISPR has also opened up exciting possibilities in agriculture. Farmers and researchers face challenges in growing crops that can withstand diseases, pests, and environmental stressors like drought and soil salinity. With CRISPR, it's now possible to enhance crops for resilience, yield, and nutritional value. Disease-resistant crops. Through gene editing, CRISPR allows scientists to make plants more resistant to diseases caused by bacteria, fungi, and viruses. For example, researchers have developed rice strains that are resistant to bacterial blight, a disease that devastates crops and reduces yields significantly. This advancement reduces the need for chemical treatments, which are costly and harmful to the environment. Improved crop yield and nutrition. CRISPR can also be used to increase the nutritional content of crops. Scientists are working on biofortified crops, such as rice with enhanced vitamin A levels, to help combat malnutrition in developing countries. Additionally, CRISPR can help boost crop yields by enhancing traits like drought tolerance, making it easier to grow food in areas affected by climate change. Reducing food waste. CRISPR enables the development of crops that last longer and spoil less quickly, reducing food waste along the supply chain. For example, CRISPR has been used to develop tomatoes that have longer shelf lives by slowing down the ripening process, which could be beneficial for both producers and consumers. 3. Environmental and conservation applications. Beyond human health and agriculture, CRISPR holds the potential for environmental conservation. As ecosystems face increasing pressure from climate change, pollution, and invasive species, gene editing offers unique tools to help manage and protect biodiversity. Endangered species. One application is in preserving endangered species. Scientists are exploring ways to use CRISPR to address genetic vulnerabilities in endangered populations. For example, certain species may be genetically susceptible to diseases, and CRISPR could help edit genes to boost immunity, making these populations more resilient. Gene drives for pest control. In certain cases, CRISPR could be used to manage invasive species. For example, gene drives, a CRISPR-based technology, can spread specific genetic traits through a population at an accelerated rate. This technique could be applied to control invasive pests, like mosquitoes that carry malaria or other vectors that disrupt local ecosystems. While this could greatly benefit conservation, it also raises ethical and ecological concerns. Reviving extinct species. CRISPR has even been discussed as a tool for de-extinction, the process of reviving extinct species. For instance, researchers are investigating the possibility of using CRISPR to edit the DNA of closely related species to bring back animals like the woolly mammoth. This is still a controversial area of research with complex ethical implications, but it highlights the transformative potential of gene editing. 4. Ethical and safety concerns surrounding CRISPR. Despite the many potential benefits of CRISPR, gene editing is not without its challenges and ethical concerns. Editing genes in living organisms, especially humans, requires careful consideration and regulation due to the following issues. Off-target effects. One of the main technical challenges of CRISPR is the risk of off-target effects, where unintended parts of the DNA are edited. While CRISPR is highly precise, there is still a risk of unintentional mutations that could have harmful effects, particularly in complex organisms like humans. Germline editing. Germline editing, or editing genes in reproductive cells, sperm, eggs, or embryos, raises significant ethical concerns. Changes made to germline cells are passed on to future generations, which means that any errors or unintended consequences could affect entire family lines. While germline editing could potentially eliminate genetic diseases, it also opens the door to controversial applications, such as selecting for physical traits or intelligence. Regulatory and ethical guidelines. Many countries have strict regulations regarding human gene editing, especially germline editing. Ethical guidelines have been established by organizations like the World Health Organization, WHO, to guide researchers in responsible and transparent practices. The ethical debate surrounding CRISPR emphasizes the need for careful oversight and international cooperation to ensure that gene editing technology is used safely and equitably. CRISPR technology is revolutionizing the field of genetic engineering with its precision and flexibility. By offering a powerful tool to edit genes across diverse fields, medicine, agriculture, and environmental science, it promises solutions to some of society's biggest challenges. The potential for treating genetic diseases, enhancing food security, and protecting biodiversity is immense. However, with great power comes the responsibility to navigate ethical and safety concerns. As CRISPR technology continues to evolve, society must carefully consider its applications and ensure that its transformative power is used responsibly and ethically to benefit humanity. What do you think of our video? Let me know in the comment section below. If you enjoyed this video, make sure to hit that like button. Also, subscribe to our channel before you go. Thank you for watching.